I would like to start by thanking the organizers for the very kind invitation to be able to talk today. So what I'm going to be telling you about today is the work of my lab that is focused on trying to understand how cell fitness is regulated during development. And specifically, we use the early mouse embryo as our model system. So one reason for this is because the early mouse embryo offers a very good paradigm to understand how cell fitness is regulated in the face of kind of very substantial changes that occur during the onset of differentiation. So cells initiate differentiation, they need to kind of completely rewire their transcriptional, epigenetic, signaling networks to be able to downregulate the expression of prepotency markers and turn on the regulation of differentiation markers in a lineage-specific manner. Accompanying these kind of changes, there's also a doubling of proliferation time that is needed really kind of to sustain the cell numbers that are needed for germ layer formation. So the large-scale dimension of these changes, what it does, it creates a large potential for let's say, the emergence of abnormal cells. And these abnormal cells really kind of need to be eliminated, not only to prevent them from contributing to further development, but also to prevent them from contributing to the germline that is specified around this period. So what this suggests is that there must be very active mechanisms of quality control that are really kind of acting to eliminate any abnormal cell that appears during this period. A few years back, we identified that right in the at the onset of gastrulation, just when differentiation is starting, embryonic differentiation is starting, there's a wave of cell death that occurs in the early post-implantation mouse embryo, suggesting that it is in this time window that this cell elimination is occurring. Furthermore, we could quantify this cell death by doing a quite simple experiment. And what we did is essentially you take embryos, you culture them in the presence of a caspase inhibitor for only 16 hours. And when we do this, what we observe is that there's a 35% increase in cell number. And what this indicates is that within quite short time window, there's a very active cell selection occurring. So if you think about the types of damage that are likely to emerge during development, there are two possible outcomes that immediately spring to mind. The first is that there's an attempt to repair this damage. But then the second possibility is that if this damage passes a certain threshold, this would lead to cell elimination. However, what I'm going to try to convince you about today is that this outcome is not necessarily predetermined and depends to a large extent in many instances on the type of damage and on the behavior of neighboring cells. So some evidence for that this context-dependent elimination occurs during development comes from when we look at the types of defect that are likely to emerge during differentiation. I told you about the changes in proliferation pattern that occur at the onset of differentiation, and these most likely bring with them karyotypic abnormalities. But karyotypic abnormalities are not necessarily directly cell lethal, because even if, for example, we take tetraploidy, so tetraploid cells survive until very late in gestation when the whole embryo is tetraploid, and these only suffer, minor pattern defects during early development. In contrast to this, a few years back, we showed that when tetraploid cells are surrounded by wild-type cells in a chimera at the onset of differentiation, this leads to cell elimination. The second type of defect that is likely to emerge at the onset of differentiation is mispatterning. But again, mispatterned cells are not cell intrinsically lethal. For example, if the whole embryo loses its ability to respond to BMP signaling, the cells don't become eliminated, but instead become mispatterned and adopt a neural fate. In contrast to this, again, a few years back, what we showed is that when cells lose the ability to respond to BMP signaling, and they are surrounded by wild-type cells in a mosaic embryo, what happens is, again, these cells are eliminated by apoptosis. And so what this suggests is that there must be some mechanism by which wild-type cells can identify defective cells and induce their elimination. And what this phenomena is very reminiscent of a mechanism that was described about 40 years ago in Drosophila and called cell competition. So what cell competition is fitness quality control, that what it does, it eliminates cells that carry defects that do not directly affect their viability when they're surrounded by other defective cells, but lead to cell elimination when they're surrounded by fitter cells. So they lead to an elimination in a context-dependent manner. To be able to gain a little bit more insight into the mechanisms underlying cell competition during early mammalian development, we have developed, in addition to the embryonic models I've just been explaining to you, we've developed embryonic stem cell models. These in vitro models in which we can essentially isolate cells that carry either a variety of different defects, such as defects in BMP signaling, tetraploidy, etc. And we can label these cells with GFP and compare their behavior when they're in a homogeneous culture to when they're co-cultured with wild-type cells. And then when we kind of analyze which kind of, let's say, which behaviors emerge specifically in heterogeneous context, we can start to kind of infer which type of behaviors are specific to competitive interactions. And so using this system, what we could show a number of years back is, again, if you take, for example, cells with effective BMP signaling, again, labeled here in green, you co-culture them with wild-type cells. These cells with defective BMP signaling are eliminated within a relatively short time window from the culture. And we can show that this elimination is really dependent on the presence of wild-type cells, because if we look at the growth of cells with defective BMP signaling when they're in a homogeneous culture, we see that they grow similar to controls. And it's specifically in the co-culture condition that these cells show a negative growth rate, indicating that they're being eliminated. Similarly, a variety of other different cell types carrying a variety of different other defects show the same behavior. So they're essentially, for example, tetraploid cells or cells with metabolic defects such as defective autophagy are all eliminated specifically when they're co-cultured with wild-type cells. What this indicates is kind of just the experiments. And then the embryo suggested is that there must be some mechanism, some common mechanism by which wild-type cells can identify defective cells and induce their elimination. So for the rest of my talk, I'm going to try and concentrate on describing the experiments that we've been doing, trying to understand essentially what are the common features that makes a cell a loser in these competitive interactions. And to start to address this question, I mean, what we did is we started by doing is we started to ask if there was a common transcription profile of the cells that were eliminated. And to really kind of to answer this, what we did is we essentially reverted to the experiment I told you at the beginning of my talk, in which we get embryos, culture them in the presence of a caspase inhibitor. And then what we do is we kind of isolate single cells, do single cell RNA-seq, and ask if the cells from embryos in which apoptosis have been blocked has a different transcriptional signature from those from control embryos. And this was work done by a very talented PhD student in the lab, who's now a postdoc, Ana Lima, in collaboration with another talented student, Gabriele Lubati, who was in Antonio Cialdone's lab in Munich, and in collaboration with Shankar Srinivas in Oxford. So when they kind of analyzed the profile of the cells of embryos treated with a caspase inhibitor, so in which apoptosis has been blocked, what they identified when they looked at the profile of these cells and clustered them according to their identity, what they identified is that in caspase inhibitor treated embryos, in addition to the, let's say, to the cell clusters that you can find in a normal embryo, corresponding to the extraembryonic tissues of the extraembryonic ectoderm and the visceral endoderm, as well as to the embryonic tissue of the epiblast, two new clusters of cells depicted here in green and in blue appeared. These kind of were specific to the caspase inhibitor treated embryos. Importantly, these two clusters of cells shared many features that were common, was essentially with the normal epiblast, but represented distinct populations. So really to kind of analyze these cells a little bit further, what kind of Gabriele went on and did is he performed the pseudo-time trajectory analysis. That is a way to kind of try and infer trajectories between cells. And when he did this, what he observed is that kind of there was a clear trajectory between, for example, the cells that you can find in the kind of normal epiblast at one end, and then with the cells that are specific to the caspase inhibitor treated embryos at the other. When he looked at the genes that would differentially express across this trajectory, what he observed is that those cells here in green were enriched for the genes that were kind of enriched in this population fell into categories of cell death and cell survival, indicating that these are most likely the cells that are normally eliminated during development. And for this reason, for the rest of my talk, I'm going to describe this cluster here as, say, the loser epiblast cluster. those cells that kind of can be found in normal embryos and caspase inhibitor-treated embryos I will describe as the normal epiblast, and the intermediate cells I will call intermediate epiblast cells. Okay. So the work that, say, my group and a number of other groups had done looking at the phenomena of cell competition in the embryo identified that kind of the loser cells eliminated by cell competition have a kind of a common set of features. For example, we had shown that kind of loser cells have elevated elevated p53 expression and lose mTOR signaling. Similarly, the group of Miguel Torres and our own group also showed that the loser cells during cell competition in mammals are marked by low c-Myc expression. When we analyze which factors have enriched targets within the genes differentially expressed in the loser epiblast, what we found is that these factors corresponded to Rictor, that's an mTOR target, to c-Myc, to p53, and to the IGF that lies upstream of the IGF receptor that lies upstream of mTOR. So essentially they presented as a typical cell competition signature, indicating that essentially this loser cluster is most likely eliminated through cell competition. When we looked in a little bit more detail at the pathways that were misregulated in these loser cells, what we saw is that they all shared a common feature. And this common feature was that they were enriched for gene signatures that were related to mitochondrial function, such as mitochondrial dysfunction or oxidative phosphorylation. So when Anna kind of looked at, for example, the expression of genes that encode for each one of the 5 complexes of that electron transport chain of the oxidative phosphorylation machinery, what she found is that these genes were all downregulated in the loser cells, suggesting that the cells that are being eliminated have mitochondrial dysfunctions in the embryo. To kind of confirm this, what Anna did is, again, what she did is she took embryos, cultured them in DMSO or in presence of a caspase inhibitor, and then dissociated single cells and analyzed the mitochondrial membrane potential of these cells. And the mitochondrial membrane potential is a feature of is a readout of mitochondrial fitness. When she did this, she observed that whilst control cells all have a kind of quite high mitochondrial membrane potential, cells from caspase inhibitor treated embryos showed a big range of mitochondrial membrane potentials, with a significant proportion of them showing a loss of mitochondrial membrane potential, confirming really our results that really kind of those cells eliminated in the embryo are most likely to show mitochondrial dysfunction. Okay. I had told you at the beginning of my talk that, for example, cells with defective BMP signaling, embryonic stem cells with defective BMP signaling or tetraploid are also eliminated by cell competition. So when Anna then, what she did is she asked if these kind of types of defect also present signs of mitochondrial dysfunction. And so when she measured the mitochondrial membrane potential in, for example, cells with defective BMP signaling or tetraploid, she also showed that these cells, she also found that these cells displayed a loss of mitochondrial membrane potential, indicating that kind of mitochondrial defects is most likely a common feature of the cells eliminated by cell competition. Okay, so these results suggested that the cells eliminated by cell competition have mitochondrial defects. So the next question that we wanted to ask is, can we manipulate mitochondrial function to trigger cell competition? So one important kind of a change that occurs at the onset of differentiation is in mitochondrial dynamics. So prior to differentiation, mitochondria in pre-implantation embryo have a rounded and a fragmented shape. And at the onset of differentiation, these mitochondria fuse and form elongated networks. This balance of mitochondrial dynamics is regulated by the relative activity of molecules that control mitochondrial fission and mitochondrial fusion. So what Anna did is she asked if disrupting these processes could trigger cell competition, and used a similar assay to the one I've described before, in which she mutates factors that are important for mitochondrial fission and mitochondrial fusion, and analyzes the behavior of cells carrying these mutations in homogeneous culture versus in eco-culture. And when she did this, she did this for a couple of genes. She analyzed the mutation of DRP1, and DRP1 is a factor that regulates mitochondrial fission, and when mutated, it leads to hyperfused mitochondria. and she also Tamzin in the lab also analyzed cells that carry mutation in mitofuscin 2. That this is a factor that regulates mitochondrial fusion, and when you delete it, essentially it leads to kind of hyperfragmented mitochondria that are really swollen. So, but when you analyze the effect of mutation, the competitive ability of cells carrying these mutations, what kind of Anna and Tamzin found is that both these types of mutations led to cells being outcompeted. So essentially, you got kind of, although these mutations did not significantly affect the growth of the cells when in a homogeneous culture, it led to their elimination specifically in co-culture. Okay, but these manipulations were, let's say, were pretty severe manipulations for mitochondrial activity. So the next thing that we did was to ask if we could do more subtle manipulations of mitochondrial activity and ask if this could trigger cell competition. And for this, she set up a collaboration with Jörg Buchstaller in Vienna and with Nick Jones at Imperial. And at this point, let me remind you that kind of mitochondria have their own genome, and the mitochondrial genome encodes for many proteins of the electron transport chain and of proteins involved in mitochondrial protein translation. Okay, so what Jörg had done a few years back is that he had isolated a number of mouse lines that all have the same nuclear background, so have a C57 black 6 nuclear background, but have mitochondrial genomes that are derived from a variety of different wild-type mice that he caught from different parts of Central Europe. And these kind of letters indicate let's say, the regions of Europe that he had caught these mice from. And the mitochondrial DNA from these mice differed from the C57 black 6 mitochondrial genome in a small number of nucleotides depicted here on the left, and an even smaller number of amino acids. So then what Anna did is she isolated embryonic stem cells from these mouse lines and asked if non-pathological changes in mitochondrial DNA sequence could trigger competition. When she did this, the first thing that she did was compare the competitive ability of cells that carry similar mitochondrial DNA variants, and she observed again that these cells would not compete. So essentially, they would grow similarly in kind of a separate and co-culture. She then went and compared if kind of two cell lines carrying mitochondrial DNAs from very distant, from mitochondrial DNAs that were very distant from the black 6 mitochondrial DNA, and asked if these kind of competed between each other. And again, she showed that they didn't. But in contrast to this, when she started to take HB cells and competed them against cells that carried a mitochondrial DNA that was more closely related to the black 6 mitochondrial genome, what she found is that the HB cells outcompeted the BG cells. So specifically in the co-culture, essentially, she got a preferential growth of the HB cells over the BG cells. And similarly, when she kind of analyzed the competitive behavior of the HB cells against the black 6 cells that have a black 6 nuclear and mitochondrial genome, she observed again that the HB cells could outcompete the C57 black 6 cells. And this outcompetition is through apoptosis, because again, if you kind of, she found that if specifically in the co-culture cells, in the co-culture, the loser cells show a large increase in apoptosis, indicating that they're actively being eliminated by the presence of, let's say, fitter mitochondrial DNA variants. So these results, what they suggest is even small non-pathological differences in mitochondrial genome can change the competitive ability of cells during differentiation. But then what we want to do is to ask if in the embryo, those cells eliminated show any difference in their mitochondrial DNA sequence. And for this, we kind of, again, talked to Gabriele and Antonio, and what they did is they looked at the mitochondrial DNA genomes of the loser cluster that are being eliminated in the embryo. When they did this, what they found is that they were enriched in sequence changes affecting the mitochondrial ribosomal RNAs, mtRNA1 and mtRNA2. So, what this indicated is that those cells eliminated in the embryo have an accumulation of mitochondrial DNA mutations, suggesting that cell competition is selecting against these mutations. Okay. So just to finish, what I'm going to kind of summarize is the way we're thinking about this is that kind of at the onset of differentiation, a variety of different defects lead to mitochondrial dysfunction, But this mitochondrial dysfunction does not directly affect viability when the cells are when the cells are surrounded by other similarly defective cells. But in contrast to this, what it does, it induces cell competition and cell elimination, specifically in kind of when they're surrounded by wild type fitter cells. One cell type one type of defect that we think is predominantly eliminated during this period of development are those cells carrying mitochondrial DNA mutations. And one possible reason for this is actually one thing I haven't mentioned, is the mitochondrial DNA genome stops replicating after fertilization and only initiates replication right at the onset of differentiation. And so it is possible that this reinitiation of mitochondrial DNA replication is error-prone, and you need cell competition to act as a purifying selection to kind of prevent these cells with mitochondrial DNA mutations from contributing to the germline. So one last thought is, I think one way of thinking about cell competition is actually that it's acting as a community effect, because what it does, it allows to perform cell replacement only when the tissue can afford to do so. So, for example, when kind of there is enough cells around to be able to effect cell replacement, then cell elimination occurs. But in contrast to this, if damage affects too many cells in the tissue, then the first, let's say, response of the cells is to be able to try and repair this response, because because essentially, if this large amount of damaged cells are removed, this may kind of compromise the survival of the embryo. And so hopefully I've acknowledged the main people who've done the work during my talk, but essentially kind of, so Anna and Gabriele performed a lot of the majority of the studies that I described, but also received a lot of help from Salva, Aida, Tamzin, Kwame, Barbara, Sarah, and Margarida over the years. And we've collaborated with the lab of Antonio Scialdone, Shankar Srinivas, Nick Jones, and George Bustaller in this work. And I'd like to thank them for that. And I'll finish there and take your questions. Thank you very much.
Thank you, Tristan. This was really a very, very interesting talk. Perhaps I might start with one very quick question, which goes back to the very beginning of your talk, where you showed that the ES cells and also embryonic cells that were at a disadvantage and were lost were the ones that had lost BMP signaling. Do you know what could be a link between losing BMP signaling and having mitochondrial dysfunction?
Yeah, that's a very good question. So we're not completely sure, but what we think is that, let's say, defects such as an inability to respond to BMP signaling may affect the ability of the cells to respond to the increased metabolic demands of differentiation. So you may need these signaling responses to be able to rewire, let's say, your metabolic machinery to adapt to the changes that occur during differentiation. Cells with defects cannot do this properly, and this is what leads to kind of them displaying these mitochondrial dysfunction, because the mitochondria either are not functioning properly because they can't perform these changes properly, or they're overworked, or let's say, or things like that. Yeah.
All right. We have a few questions coming up from the floor, so I'll just read a few. We have one from an anonymous attendee that asks, how do you exactly define fitness in this cell competition context?
A very good question again. So we define fitness as the ability to survive in a heterogeneous environment. So the fitter cell is the one that essentially, when placed in a competitive environment, survives. We think what determines cell fitness ultimately is mitochondrial activity, but let's say the ultimate readout is this ability to survive in a heterogeneous environment.
And have you looked at any intercommunication pathways that may regulate or control the interaction of mitochondria and nucleus genes?
We are looking. So far, we've not been able to pinpoint any specific pathways, but certainly we're kind of giving a little bit of attention to, for example, signaling pathways that are activated by mitochondrial DNAs, such as the innate immune type responses, and trying to look if they can be activated differentially between winner and loser cells.
We also have a question from William Hill who says, great talk. And oh, the chat just moved and I lost it. There we go. How do you think loser mitochondrial defective cells are recognized by the wild type neighbors? Is this concern between different mitochondrial defects?
Oh, yeah. Again, a very good question, and we don't know. That's a quick answer. I think ultimately we're thinking about one of two possibilities. Either it affects the ability of the cell to essentially, there's a competition for nutrients, and the mitochondrial defects affect the ability of the cells to uptake the nutrients when surrounded by other cells that can uptake these nutrients more efficiently. That's one possibility. The other is that the mitochondrial DNA changes elicit a signaling response that triggers, let's say, a recognition pathway to be activated in winner cells. And this may be, again, as I suggested, maybe through an innate like immune pathway that can be triggered by mitochondrial DNAs.
I think I might have time for one, maybe two more questions. Let's see how efficient we can be. One question from Rocio Sancho in the audience, who says, in the cell competition experiments, I guess there must be a minimum number of normal cells able to compete out or induce death in a specific aberrant cell. So one could assume that in a cluster of mutant cells surrounded by wild-type cells, this competition pressure would be lost, and at least the cells in the middle, which are surrounded by mutant cells, may still be fine. Is that the case? Is it possible, essentially, to escape the competition pressure?
Yes, it is. Yeah, so definitely that's spot on. So essentially, when you're in a scenario in which your neighbors are preferentially of a similar, let's say, defective status, you can avoid cell competition.
But I guess how effective is that? Would you have that first the peripheral cells that are mutated are eliminated, and then the ones that are more central now have wild-type neighbors, and then they get eliminated next? So do you see a full elimination of the mutant clone? Or?
Again, very good question. It's dependent. Some clones are not fully eliminated ever. So suggesting that they may be, as well as let's say the behavior of the neighbors, there may be, let's say, intrinsic factors that allow them to kind of avoid being outcompeted.
Perhaps we have time for one. We have a lot of questions. I hope that you can see them separately, and perhaps you can reach out to a few people on the side. I'll come up with one last one from Michel Perchard. Just wondering if you have manipulated the OXPHOS pathway or other aspect of mitochondria to make them super competitors or more successful, or do they just always get less successful as soon as you mess up with the mitochondria?
Have we been able, well, the HB cells that carry these mitochondrial DNA changes, they do outcompete C57 black 6 cells that don't carry any specific mutation. So whatever that is present in the HB mitochondrial DNA allows them to increase the fitness. And that's one thing we're trying to look at. It's essentially what is specific about the changes that these cells have that allow them to outcompete cells that are, let's say, the black 6 cells that are supposedly perfectly normal.